Good afternoon. So today I'll be talking about TrueAllele interpretation of DNA mixtures. And unlike when I first spoke here about 3 years ago, we have a series of cases that we've done with the county, and so there's more to talk about. What I'd like to do is to first begin for the first half hour and tell you about DNA and mixtures and what the issues are and how do you know that it's reliable. And then about a half hour in, we'll start with some cases. Okay, so with ordinary DNA evidence, you have one person. The smiley face represents a person. And that person has maybe a trillion or more cells that have nuclei in them. Each nucleus has 23 pairs of chromosomes, one inherited, each copy from each parent. Here we're taking a look at a chromosome, and if you look at a particular location on one of these chromosomes, that's a genetic locus that's used as a marker in forensics for identifying people. You'll hear a lot more about that over the next 2 hours. But what's the content of what's sitting at that locus is a pair of genes or alleles. It's the value of what is in that DNA sentence at that particular location. And so here, just as an example, imagine that what a person has inherited is one allele, one sentence of length 7 from one parent. And here you see a longer length, and where these lengths come from I'll tell you about later, of length 14 from another parent. And so their genotype at this location for that person on that chromosome would be a 714. And when DNA data is developed by a crime lab at, say, 15 different locations, and this is one of them, what they would see is a graph called an electropherogram. And what is on the electropherogram on the x-axis is the size. So you see that 14 is longer than 7. It goes from left to right in terms of size. And the height is in fluorescent units, and it expresses the quantity. And the other thing you can see is this is pretty easy. Right? If all you had was single source DNA and you saw at this one of 15 locations that there was a peak at 7 and a peak at 14, that person's genotype would be a 714. And that's what you have with reference samples and very easy DNA evidence, and there wouldn't be much of an issue. But that's not what the talk's about today. So imagine you have two people in the evidence. Then there are two genotypes. In addition to the first person whose genetic location contributed a genotype, two alleles, a 7 and a 14, there could be a second person. And that person also has DNA that's contributed to the biological evidence. And they may have the same or different values. Here, for simplicity, I drew them as the same. Imagine that their genetic type is a 10 and a 12. And again, in forensics, the numbers correspond to how long the DNA fragments are because of repeated elements that I'll talk about later. So now instead of just having one genotype with two alleles from one person or a second genotype with two alleles from one person, you have two people with 4 alleles, and the signal gets more complex. And this is what a DNA mixture looks like. Mixtures can have contributors from 2, 3, 4, 5, 6, however many people detectably contributed to that biological sample. And this pattern is a little more complex. You can't just look at it and say with absolute certainty that I know of the 100 possible allele pairs that could exist, I definitely know that it's a 7 and a 14. With a computer, you might be able to do that, but we'll get into that. So this is what a mixture looks like. And what's important about it are at least 3 things. The first is the peak heights make a difference. The fact that the 7 is taller than the 10 is meaningful because the height of the peak corresponds to the quantity of DNA. So if there's more of a 714 individual than a 1012 individual, then the peak heights with random variation accounted for should be taller. Secondly, it's the pattern that matters. You see 2 tall peaks, 2 short peaks, and that's important in interpreting evidence. It's not just there's a peak present or not. It's their heights and the pattern. And also, this height information and the pattern can give a computer information about the variation in the experiment. Okay, so that's what mixture data looks like. This is what human review does. And I'm going to describe a procedure. As far as I know, this procedure has never been validated. There's been no papers written about why it works, how often it gets the answer right, how often it gets it wrong, or how reproducible it is. But this is the procedure that's done in most crime labs today. It's called, we'll give it a name, it's called the Common Probability of Inclusion, or CPI. That's what it's referred to in the field as CPI. And what it involves is taking a threshold and not trying to interpret the data by trying out all possibilities, but taking a surface look at the features. So here's some peaks, some tall, some low. A threshold is determined as a one-size-fits-all for the laboratory. And then a line is drawn across the quantitative data. Peaks that are over the threshold are considered to be in. Peaks that are under would be considered to be out. Now, you can look at the data and see this doesn't quite look right. It looks like there's one guy who's a 714 and somebody else who's a 1012. And you're right. But this is a simplified method to take a complex problem and reduce it to something that a person can do in, a few hours. So a line is drawn, and the all or none alleles that result from the thresholds, if you were to list all possible pairings, which is what this method does implicitly, there could be a 7 and a 7, a 7 and an 8, a 7 and a 10, a 7 and a 12, and so on. There'd be 4 possible pairings, and this interpretation method of the mixture data that people do assigns equal likelihood to all 10 possibilities. Now, if some of these are far more likely than others, like there's a major contributor that's a 714 and a minor corresponding to the smaller peaks that's a 1012, you could be losing a lot of information. And in fact, that's what happens with these methods. You lose a lot of information. There's a study we've recently done with Virginia Department of Forensic Sciences that I'm going to be talking about as I'm describing the reliability of these different methods. This is CPI. So this is a study. I'll describe where the data came from in context in about 5 minutes. This is a study where we looked at about 100 items of evidence and the match information that was obtained. On those 100 items, about 3/4 of them could get a score using this method called CPI that's been in use for about 15 years in the US and around the world. What I've shown on the x-axis is the amount of information that the match statistic got. The way scientists count information is by the number of zeros in something like a match statistic. So a number, 1,000 is a 1 followed by 3 zeros, so its information is 3. A million is a 1 followed by 6 zeros, so its information is 6. Right? 9 corresponds to a billion, 12 corresponds to a trillion. It also goes negative, but I'll tell you about that later. So here we see on a scale of information that ranges from 0 up to 25 that CPI's information over these 100 mixtures, accurately, or at least giving some match statistic for most of them, is centered in some region, and the number of zeros, which is what I'm showing over here, is 6. So the average match statistic is 1 million. It can go up to 9 and give a billion. It can go down to 3 and give 1,000. But that's the average match statistic. It's in the millions. And that's, if you do DNA cases, that's what you probably have been used to up until 2 or 3 years ago. The match statistics for mixtures always came back around 1 million. And this is on almost 100 items showing that, in fact, that's where the numbers are. They don't go up to what a single source would be of DNA from one person. They don't go down to 0, which would be no information. They're clustered around 1 million. And that was the status quo for about 15 years. Then there were concerns about the reliability of these methods, and so the FBI wrote a paper in 2009 describing what it did, and this was adopted by the Scientific Working Group on DNA Analysis Methods, abbreviated SWGDAM, and in 2010 they published new guidelines that most labs in the country began to follow. And they essentially raised the threshold for human review. they introduced a new constraint. It was not enough that the peaks had to be over a threshold. The threshold is now about 3 times higher, but any peaks that were seen at the lower threshold also had to be seen at this higher threshold. Otherwise, that locus could not be used in the match statistic. And so the result was a lot of data like this was thrown out in the sense that it was not used in the match statistic. And that resulted in a precipitous drop in DNA match statistics. And in our study with Virginia, we see that. This was the old method, CPI, which used a lower threshold. And now the newer method, modified CPI, that used a threshold about 3 times higher in each lab, resulted in a lot less information. Instead of having 6 zeros for 1,000,000 as the average match statistic with CPI, instead the number of zeros averaged 2, and the match statistic was now in the hundreds, which is not the most persuasive evidence you can present, right? And what also happened is that about half of the items were just called inconclusive entirely and not used with a match statistic. Now, since match statistics are required for biological evidence to be introduced in many jurisdictions, this led to less DNA evidence being presented in court from the same number of samples that were brought into the laboratory, processed, analyzed, and produced data. Okay, so that's the red shift to the left. Okay, the modified CPI on the same data lost a lot of information, averaging around 2 zeros instead of 6. The SWGDAM guidelines, though, did have another paragraph in them, paragraph 3.2.2, which said that if you're not using stochastic thresholds, there's a way out. You can use a computer instead, a validated computer that figures out the probabilities of genotypes. And that's in the language. And so Virginia opted to use TrueAllele Casework for their DNA mixture statistics as a way of computing this on the same data. This is what the system looks like. There's a user interface where people happily sit in front of it, entering data, asking the computer to solve problems. The reason they're happy is the computer goes off and solves all the problems in a way I'll show you, does all the heavy lifting and the hard work, and then they can come, when the computer's done, they can look at the answers on their screen again. With parallel computers, you can solve a lot of problems at once. At our office in Oakland, our computers can work on about 100 mixture problems at the same time, each on a different processor. Does this work? I'm going to be talking more about that, but the answer is yes. TrueAllele is the most validated method of interpreting DNA mixtures. There have been over 20 studies done. 5 of them have been published in peer-reviewed journals, and they analyze different aspects of them. The newer papers, the newer studies that we're doing go out to 5-person mixtures, look at what happens when there are relatives, just ask question after question with challenging sample, and these are done by other laboratories as well as by Cybergenetics. This TrueAllele reinterpretation was described in the Washington Post 2.5 years ago under the title, Virginia reevaluates DNA evidence in 375 cases. We'll get back to that number 375 at the end, but in the article they noted that police and DNA experts find mixtures a challenging endeavor, with some experts saying if you ask 10 different analysts what the answer is, you'll get 10 different answers. Okay, so this is what we did in this study. This study is based on the reports that we actually submitted to court. These are reports that Cybergenetics prepared, that I reviewed, signed, and submitted as working with the lab to extend what the lab was doing in a number of cases. There were about 150. There were 72 criminal cases where reports were filed that involved 92 evidence items. And because a mixture can contain more than one person who you would match, it involved over 100 genotype comparisons, 111. And the criminal offenses were very broad range. Some of the more common ones were homicide, robbery, weapons violations, and sexual assault. So what does TrueAllele do? What does the computer do that's different from what a person does? There's several things. First of all, it uses the quantitative information, and it tries to figure out not just the surface features of what the peaks look like, but it tries to work out the genotype, that genetic type, like the 714 or the 1012, the allele pair, of every contributor to the mixture. So it separates the mixture. In the example that I'm showing, which came from a case in Virginia, there was a 75% component that corresponded to the victim, and a 25% component that corresponded to the perpetrator. And this is just a scale from 0 to 100 that's showing how much the contribution is. And TrueAllele separates the components. If there were 3 contributors, you'd see 3 peaks here, and those numbers would add up to 100%. So it separates out the genotypes. And the way it infers those genotypes is in a way only a computer can do. What it does is it doesn't really focus too much on the data when it gets started. It focuses on trying out every possibility. This is a good possibility because it explains the data well that you see in the picture, where there's a lot of a 7 and a 14 as the pair of alleles from one individual, and then maybe half as much signal from shown in green as a pair of rectangles from a second person. That's a 10 and a 12. And by adding these rectangles together and accounting for other artifacts and variations in the DNA experiments in the lab, the computer compares the patterns that it proposes against the data. This pattern, a lot of 714 and a little bit of 1012, explains this data very well. So it has higher likelihood. But the computer's moving the rectangles everywhere. It's trying out an 8 and a 19 in different amounts, different heights. Most patterns don't explain the data very well, and they get less likelihood, and those genotypes become less probable. Some patterns explain the data better, and those explanations confer higher probability to their genotypes. And the computer can do this 10 or 100,000 times, trying out pattern after pattern at each of 10 or 20 different locations, trying to explain the mixture through a random search process. And when it's done, in this particular case, it placed most of the probability, 98%, on the genotype, the allele pair 1012, for that minor contributor shown in green, which intuitively is what you see in the data. It's what a person would think, but it's not the procedure that crime labs follow. So now we can ask, how reliable is this method? Reliability has 3 basic components. One is the extent to which this interpretation of the data identifies the correct person. That's sometimes called how sensitive the method is, or sensitivity. If TrueAllele is looking at the data, how often and to what extent does it make true DNA mixture inclusions? In this study, out of the 111 comparisons that were made, it found 101 genotype matches. So it was reasonably sensitive in finding over 90% of what was there, and it gave match statistics that could be appreciable. 82 of those had a match statistic over 1 million. This is the range of the results. So going from 0, which is no zeros after the 1 or just 1, up to 24, which is 12 plus 12 zeros after the 1, or a trillion, that's the match statistic. TrueAllele fills the entire range of match statistics. And that's what you would expect. If you have random amounts of DNA in random mixture quantities, then you would expect to see the full range between no information and the full information if you had a single source individual and with almost not even a mixture anymore. That's what you would expect, and we'll get back to it later, but that's actually what you see. It fills the whole range. So the method is sensitive. It's detecting information that's there. Then there's a second question. Is it overcalling? Is it making mistakes? Does the interpretation misidentify and get the wrong person. That's asking how specific the method is, or specificity. And so in order to study this, we took the 101 matching genotypes and compared them against 10,000 randomly generated genotypes. None of these are expected to be contained in the actual mixture because there's on the order of a trillion possible genotypes, and if we're just kind of looking at tens of thousands of them randomly, those are not going to be the genotypes that are in the actual evidence. We did it for 3 different ethnic groups because that's customary in forensics. And the result of comparing 100 genotypes against 10,000 random references is over 1 million comparisons. And this is what we found. Here, 0 is now all the way on the right. And what we found were negative numbers. So what's negative? Negative 6 means 1 over 1 million. Negative 12 means 1 over 1 trillion. So unlike the CPI method, which can only include people, it's called the probability of inclusion, likelihood ratio methods like TrueAllele can both include with numbers to the right and exclude and show that somebody's not present in DNA evidence, or at least there's no scientific support for it, with numbers that go as 1 over 1 million, 1 over 1 trillion. In this case, negative 19. 18 is 9 plus 9. There are 9 zeros in a billion. So that's 1 over 1 billion was the average exclusionary power. And it's described in a report, but there were very few false positives, maybe at the rate of 1 in 20,000, that even went up to a number like 10 or 100. So the method's very specific. It's also reproducible. If I have a random process on a computer and I run it twice, do I get a match statistic like 1,000 the first time and 1 trillion the second time? Or if I run it twice, do I get a number like a trillion the first time and then around 1 trillion the second time? The reason you need these to run a random process is that in a particular mixture problem, there can be hundreds of variables, each with their own equations. And to solve them, the only way that people know how to solve them using computers is by random search algorithms that have been around since 1950 or so. They're very well established in most fields of science and social science. So in order to check that sampling variation of how the computer solves the problem, we compared that. The x-axis is for the match statistic on the first computer run. The y-axis is for the match statistic on the second computer run. If the numbers were absolutely identical, they would all line up at 45 degrees where y equals x. And the main thing to notice is you're not seeing points very far away from that line, y equals x. They're very much concentrated on that line. And you can compute a statistic to ask what's the variation within group, and that corresponds in logarithmic units or exponents to 0.3, which is about a factor of 2. So on average, the answers are quite reproducible within a factor of 2 as a standard deviation. Okay, so let's compare these methods. Here's the old CPI method that had been enjoyed use for maybe 10, 15 years. It has an average statistic of 1 million, 6 zeros, and it can interpret most of the evidence. This is the newer method that the community is now using starting 4 years ago, and its average match statistic is only 2 zeros in the hundreds, and it can't analyze or give an interpretation for at least half of the mixture evidence that's coming in. It's just thrown away. You have the signals, but there's nothing you can do with it. And this is TrueAllele. TrueAllele spans the spectrum as one would expect from no information to full single source information, and has an average value measured around 100 billion, or 11 zeros. It's just in the middle of the range. And possibly most importantly, it produces results for over 90% of the mixtures. So no DNA is left behind, right? Nothing is thrown out. It's being used. You can actually turn this level of concentration and the range into a measure of accuracy. If you keep increasing the information as you move to left to right, the area under that curve is a line. And so you'd expect with this black line that that's what a method that's accurate would look like over 100 different matches. In blue, we see that's what TrueAllele actually is, and that's statistical support that it's an accurate method. In green, we see CPI, and it does not look like that black line. It's shifted to the left. And MCPI, that throws out much more information, is shifted very far to the left. And statistically, you can show, based on this concept, that the full range of 100 matches should fill the spectrum. It fails that test, and that's support that perhaps these methods are not that accurate. They also don't correlate very much with information, as we also saw in the study. Okay, so what happened in Virginia? 144 cases were analyzed by computer. We were asked to generate 72 case reports. There were 10 trials. So far, there are more. I testified at 8 of them. In brown are 2 trials where an analyst from the government lab in Richmond testified. And you'll be hearing more about how you testify about these results for most of the remainder of this presentation. And as you can see, the offenses span the range of weapons, bank robbery, rape, homicide, and so on. There's a full range of offenses in the study. And actually, in this particular set, whenever TrueAllele was used in court, somebody went to court, they received some sort of sentence. Most of the cases resulted in pleas when there was an outcome, in the sense that by having strong DNA results, whereas before you didn't have any at all, a plea bargain could be obtained. That was some sort of compromise. So where's TrueAllele today? 20 years ago, we first invented the math and the algorithms, developed the computer systems. There are now about 10 laboratories that we're supporting that have users and are developing various workflows. There are 3 labs that are now using TrueAllele routinely in casework. Virginia just came on board writing its reports, I think, about a month ago. There's a lab in California using a very automated workflow where the data goes into the computer, matches occur on a local database, the answers come back, then they just harvest the results without spending inordinate time up front looking at the data. They look at the answers first and then go back and look at the data. There have been many validation studies done, and we've been giving a lot of talks. There are 50 of them on YouTube that you can see. I'll give you the link later on. We've been training hundreds of students, mainly forensic analysts. There have been, I think, 4 admissibility hearings so far. There are another 4 coming up in the next half year. There was been about 20 trials in which people have testified about TrueAllele results. There have been thousands of people who've heard talks about it, for better or worse, and there have been almost 200 case reports filed. In Pennsylvania, there's been interest in TrueAllele, most of these with crimes and case reports filed in over 10 counties across the state. And in Allegheny County, there have been 16 cases so far with 12 case reports. I'm not counting multiple reports in one case. There have been 3 trials, and one of the first results was actually an exoneration that resulted from a study that we were doing with a crime lab. The crimes are typically violent crimes. TrueAllele is probably most often used for rape and homicide offenses. The evidence is a wide range of evidence. Notice that it's not, these are not intimate samples. This is clothing, guns, fingernails, shotgun shells. These are the various defendants who've had TrueAllele reports. The outcome in the 3 trials so far were guilty. These are the sentences. And as I said before, most of the time there's a plea. It doesn't go to trial, because whereas before there was DNA evidence, but then because of the lack of a match statistic, there was no DNA evidence. And so now you're trying to go to court without DNA. What the computer does is it accurately states the match statistic of the information that's actually there that connects crime scene evidence with an individual.